The basic question we sought to address in my presentation is, first of all, is there a shared or public neoantigen that results from mutant PIK3CA, again, a driver oncogene that is commonly mutated in prevalent solid cancers like breast and endometrial cancer? Our strategy was really twofold. One was first to do an immunopeptidomics screen in which we intentionally electroporated into an artificial antigen presenting cell the full-length gene sequences for common mutant PIK3CA variants alongside the most common HLA types that are seen in North America as well as in Western Europe. And we then did mass spectrometry to see if any of those combinations generates a naturally processed and presented shared neoantigen resulting from mutant PI3 kinase. We developed one candidate that is compatible with HLA-A3. This is a tissue type that is about 20 to 28% depending on the specific region, both in North America as well as Western Europe, so it is quite common. And we showed that this epitope is in fact immunogenic in a subset of cancer patients who have the right HLA type and mutant PI3 kinase. Interestingly, about 1 patient in 3 develops a natural or spontaneous immune response to this target. Why it's not 100% is a question we're trying to address right now. But those data show that this epitope, which we identified, can be immunogenic in cancer patients. We went on to clone a series of unique gene sequences that encode T-cell receptors, the natural antigen receptor that's on all T cells that can recognize this epitope. And we found one TCR in particular, which we've termed TCR4, which has some very interesting and unique properties. One of these properties is that its CDR3 beta length, the loop emanating from the TCR that actually contacts the peptide, was extraordinarily long. In fact, it was among the longest CDR3 beta loops we've seen among over 17,000 TCRs that can recognize peptides that are bound to HLA A3 and A11. We went on with a fantastic collaboration with a structural biology team to do X-ray crystallography of TCR4 bound to its target in addition to other TCR candidates, and found that this long loop actually was functionally significant. It basically formed a curtain around most of the bound peptide, and functionally this meant that this TCR was exquisitely specific for this particular peptide. So if we mutated and altered the peptide through either alanine or glycine substitution, the TCR completely lost function. So this implies that this TCR has a very, very low risk of cross-reactivity against undesirable peptides, a property that is, I think, very important for developing a TCR therapeutic. And finally, in a preclinical animal model, we showed that we could see tumor regression, specifically of tumor cell lines that naturally express HLA-A3 and the mutant form of PI3 kinase, but not a line that expresses A3 in the wild-type form of PI3 kinase. So based on this encouraging potency and safety profile, we're now developing a first-in-human clinical trial based on this TCR, which hopefully will begin in the next 18 months or so.